All right, good afternoon, everyone. My name is Dr. Eric Toring. I'm a part of the Executive Vice President team with the American College of Veterinary Preventive Medicine. And I want to welcome you today to our continuing education webinar. I want to welcome our active ACVPM diplomates and active members of our partner organizations, the American Association of Food Safety and Public Health Veterinarians, the National Association of Federal Veterinarians, and the U.S. Animal Health Association. This webinar has received 1 hour of RACE-approved continuing education. To earn that continuing education credit, an attendee must attend for a minimum of 50 minutes. CE certificates will be emailed to attendees within 7 to 10 days. We will, after the presentation, take questions and answers. We'll take questions, we'll give answers. But if you have a question regarding this webinar, please type your question in the Q&A feature of Zoom. With that, I will turn this over to Dr. Suzanne Tomasi, who is our American College of Veterinary Preventive Medicine Continuing Education incoming chairperson. Suzanne?
Thank you. And welcome, everyone. I would like to introduce Dr. Megan Nichols and Dr. Michelle Waltenberg. Dr. Nichols serves as the lead for the team at CDC that investigates multi-state outbreaks of Salmonella and E. coli resulting from exposure to animals, pet products, and raw milk. Prior to joining the CDC, Dr. Nichols worked in her home state at the New Mexico Department of Health for 5 years. She received a Bachelor's of Science degree in Animal Science from New Mexico State University, a Doctor of Veterinary Medicine from Colorado State University, and a Master's of Public Health in Food Safety and Biosecurity from the University of Minnesota. Her areas of interest include zoonotic diseases, food safety, and pediatric health. Dr. Michelle Waltenberg is a veterinary epidemiologist at CDC. Prior to joining CDC, Dr. Waltenberg worked as a small animal veterinarian in Illinois. She received a Bachelor's of Science degree in microbiology from the University of Miami and a Doctor of Veterinary Medicine and Master's of Public Health from the University of Illinois. Her areas of interest include zoonotic diseases, One Health, and antimicrobial resistance. Thank you, everyone, and welcome Dr. Waltenberg and Dr. Nichols.
Good afternoon, and thank you so much for the opportunity to be here today. So to start, and for today's audience, I want you to imagine that you're a veterinarian practicing at a veterinary specialty clinic. And I know for many of us, this isn't necessarily a stretch. Your clinic sees mostly companion animals and has both surgical and medical services. You receive a phone call from the Veterinary Diagnostic Laboratory, which you send organisms to routinely for bacterial culture. And the diagnostician on the other end of the phone notes that E. coli isolated from one of your patient's dog's wound culture has some antibiotic resistance, and they wanted to check in. Indeed, they had recently added imipenem to their antibiotic susceptibility testing panel and have identified this isolate as resistant. So go ahead and open up the Q&A and ask the attending veterinarian on the phone with this diagnostician, what questions do you have? Are there any additional tests that you might want to run? Excellent. So I see one. Where does the owner work and what is their occupation? That's an excellent question. Any others? How common is imipenem resistance? Also an excellent question. And what has the patient been treated with already? So what if I tell you if this is a patient that has pretty significant skin conditions and the patient has been referred to your practice, you've been working with this particular patient for over 8 months and they had been to a couple referral veterinarians prior to seeing you. Travel, excellent question. I might also note that maybe the patient has been on antibiotics before, a series of antibiotics for different skin ailments and treatment for allergies. Is it a service animal? Excellent question. So I'll add an additional, there's a PCR comment. Excellent. I think additional testing. So Enterobacterales, like E. coli, that test and are resistant to at least one of the carbapenem antibiotics, including imipenem, ertapenem, meropenem, or doropenem, or those that produce carbapenemase, which is an enzyme that can make these organisms resistant to carbapenem antibiotics, are often called CRE. And the carbapenemases, these enzymes that might be present, can inactivate carbapenems and other beta-lactam antibiotics, including penicillins and cephalosporins. So the presence of a carbapenemase is usually sufficient to cause carbapenem resistant. So based on your request, this veterinary diagnostic laboratory goes ahead with submitting the isolate for whole genome sequencing. And maybe they actually work with their local public health laboratory or with one of their federal partners to do this. And they sequence the E. coli isolate and identify that the organism does have the gene that produces carbapenemase, or that indicates that enzyme is able to be produced. And it has some metallo-beta-lactamase genes. And the organism is identified as a E. coli New Delhi metallo-beta-lactamase 5. If you're told this as a veterinarian, do you know if this is reportable in your state? What if you subsequently find out, because you know your local public health veterinarian and you call them, that NDM5 E. coli are organisms for which CDC recommends a public health response to even a single case found in a human healthcare facility in order to prevent its spread? Thus, rapid response, including laboratory testing, is often needed to conduct case finding and to prevent any additional spread in either human healthcare or veterinary healthcare settings. Well, now that I've got you started thinking, I'll actually turn it over to my colleague, Dr. Waltenberg, to further describe a survey done to actually address these and other questions regarding testing of CRE, or testing for CRE, in veterinary diagnostic laboratories in the United States. Over to you.
Awesome. Thanks so much, Megan. Just confirming that you can see my screen.
We can.
Perfect. Thank you. So as you all just heard, carbapenem-resistant organisms, or CROs, are a significant threat to both animal and human health. This afternoon, I'll be sharing the results of a national survey to better understand the capacity of veterinary diagnostic laboratories, or VDLs, to detect these organisms. Carbapenems are antimicrobial drugs reserved for the treatment of severe multidrug-resistant gram-negative bacterial infections in hospitalized patients. The emergence and spread of CROs, including carbapenem-resistant Enterobacterales, or CRE, carbapenem-resistant Acinetobacter baumannii, and carbapenem-resistant Pseudomonas aeruginosa, are a major public health concern because they can cause infections that are challenging to treat and are associated with high mortality and high healthcare costs. There are several mechanisms of carbapenem resistance, but carbapenemase producing strains have been categorized as an urgent public health threat because they can rapidly spread multi-drug resistance between different bacterial species. Correspondingly, CROs have been made reportable to public health in many jurisdictions, though reporting requirements vary by jurisdiction in terms of organism, resistance, and mechanism. Although carbapenem antibiotics are not labeled for use in animals in the United States, among the limited reports available, carbapenems have been used in veterinary medicine to treat infections refractory to standard antimicrobials. CROs have been documented in companion animals, i.e. dogs and cats, but the overall incidence of CROs from companion animals in the United States is unknown. Recent reports in the United States and abroad have documented single cases and large outbreaks of CROs among companion animals. In some instances, transmission between people and animals has been documented. Healthcare-associated spread among companion animals has also been documented in veterinary hospitals. To date, CRO surveillance in the United States has been primarily focused on human healthcare settings. However, antimicrobial resistance is not confined to human medicine, and public health is increasingly recognizing the importance of the environment and animals in the emergence of resistance. CRO surveillance in companion animals in the United States is important because of the potential impact on both human and animal health. However, the capacity of US VDLs to detect CROs is unknown. The ability to detect these antibiotic-resistant pathogens and conduct public health response might be limited if VDLs cannot quickly identify resistant bacteria. We assessed the ability of VDLs to detect these high concern organisms from isolates from companion animals. Our first objective was to assess current activities and technologies used to detect carbapenem resistance in Enterobacterales, Pseudomonas aeruginosa, and Acinetobacter baumannii from companion animals, and determine how information regarding carbapenem resistance is shared with clients and stakeholders. We asked laboratories that were part of one or more of the following networks to participate: the American Association of Veterinary Laboratory Diagnosticians, the USDA Animal and Plant Health Inspection Service National Animal Health Laboratory Network, and FDA CVM's Veterinary Laboratory Investigation and Response Network. A 54-question survey was jointly developed by experts in veterinary zoonoses, antimicrobial resistance, healthcare-associated infections in humans, and clinical microbiology using research electronic data capture. Questions were limited to practice about companion animal or dog and cat isolates, including bacterial identification, antimicrobial susceptibility testing or AST, carbapenem susceptibility testing, and carbapenem resistance mechanism testing. We also asked about actions initiated when carbapenem resistance is detected. And we also collected some laboratory test results from January 1st, 2019 through June 30th, 2020, including the approximate number of Enterobacterales, Pseudomonas aeruginosa, and Acinetobacter baumannii isolates from companion animals tested for antimicrobial susceptibility and the number resistant to a carbapenem. AAVLD, Nuln, and VetLearn distributed the survey electronically to their member laboratories on December 7th, 2020, requesting survey completion within 5 weeks. Among the estimated 74 VDLs in 42 states sent the survey, 23 or 31% of laboratories from 22 states completed the survey, with locations of survey respondents displayed on the map. No geographic clustering was noted among survey respondents, and all US Census regions were represented. Sources of clinical specimens for the 23 laboratories surveyed included academic and private veterinary practices, other VDLs, zoos and/or aquaria, wildlife agencies, research companies, and livestock producers. The proportion of specimens received from companion animals relative to other animal species varied by laboratory and ranged from less than 10% to greater than or equal to 75%. At 9 reporting laboratories, companion animals were the source of at least half of specimens submitted. Testing standards and methods varied by laboratory, with the majority reporting using a combination of Clinical and Laboratory Standards Institute, or CLSI M100, and CLSI Vet08. 16 or 70% of VDLs reported using multiple methods for gram-negative species identification, of which the most common was multidrug-resistant. All 23 laboratories surveyed reported conducting AST on gram-negative isolates from companion animals. And of these, all 23 reported conducting AST on Enterobacterales isolates, 22 or 96% reported conducting AST on Pseudomonas aeruginosa isolates, and 21 or 91% reported conducting AST on Acinetobacter baumannii isolates. Of the 23 labs that reported performing primary AST on Enterobacterales isolates, 21 or 91% reported including at least one carbapenem. Of these, all labs reported including imipenem, and 1 lab also reported including meropenem. Of the 9 labs that reported performing secondary AST on Enterobacterales isolates, 6 or 67% reported including at least 1 carbapenem on this panel. Of the 22 labs that reported performing primary AST on Pseudomonas aeruginosa isolates, 19 or 86% reported including at least 1 carbapenem, and of these, all reported including imipenem. Of the 6 labs that reported performing secondary AST on Pseudomonas aeruginosa isolates, 4 or 67% reported including at least 1 carbapenem on this panel. Of the 21 labs that reported performing primary AST on Acinetobacter baumannii isolates, 19 or 91% reported including at least one carbapenem, and of these, 17 or 89% reported including imipenem, and one lab each reported including meropenem and doropenem. Of the 6 labs that reported performing secondary AST on Acinetobacter baumannii isolates, 3 or 50% reported including at least one carbapenem. Of the 23 labs surveyed that reported performing AST on Enterobacterales isolates from companion animals, 21 reported the estimated number of isolates that were tested for antibiotic susceptibility and or the number resistant to a carbapenem. From January 1st, 2019 through June 30th, 2020, several thousand Enterobacterales isolates were tested for antibiotic susceptibility across the 21 laboratories that provided estimates. Of these, 20 laboratories reported the estimated number of Enterobacterales isolates from companion animals that were resistant to a carbapenem. As shown on the graph, 14 labs did not identify any carbapenem-resistant isolates. 3 estimated 1 to 5 isolates were resistant, 1 estimated 11 to 15 isolates were resistant, and 2 estimated that greater than 16 isolates were resistant to a carbapenem during this time frame. These findings indicate that at least 46 CRE isolates were isolated at 6 laboratories surveyed during the time period of interest. Of the 22 laboratories surveyed that reported performing AST on Pseudomonas aeruginosa isolates from companion animals, 20 reported the estimated number that were tested for antibiotic susceptibility and or the number that were resistant to a carbapenem. And again, during the same time period, January 1st, 2019 through June 30th, 2020, several hundred Pseudomonas aeruginosa isolates were tested for antibiotic susceptibility across the laboratories that provided estimates. As shown on the graph, 18 labs provided estimates for the number of isolates resistant to a carbapenem. 12 did not identify any carbapenem-resistant isolates. 4 estimated 1 to 5 isolates were resistant. 1 estimated 6 to 10 isolates were resistant, and 1 estimated 11 to 15 isolates were resistant to a carbapenem during this time frame. Of the 21 laboratories surveyed that reported performing AST on Acinetobacter baumannii isolates, 16 reported the estimated number that were resistant to a carbapenem. 15 or 94% did not identify any carbapenem-resistant isolates, and 1 estimated that 1 to 5 isolates were resistant to a carbapenem between January 1st, 2019 through June 30th, 2020. We also asked questions about what, if any, additional laboratory testing would be performed if carbapenem resistance was detected from a companion animal. 9 of 23, or 39% of labs, reported performing additional tests to identify carbapenemase production or carbapenemase genes. As displayed in the figure, of these 9 labs, 8 reported performing whole genome sequencing, 2 reported performing polymerase chain reaction for carbapenemase genes, and 1 lab each reported performing phenotypic testing or commercial molecular tests. We also asked questions about what actions would be initiated if carbapenem resistance was detected in an isolate from a companion animal. And as you can see from the figure, VDLs reported they would initiate a variety of actions if a CRO was detected from a companion animal. 57% reported notifying the submitting facility clinician, biosecurity officer, or infection prevention staff. 35% of VDLs across 8 states reported that they would notify public health if carbapenem resistance was detected. And these actions included either notifying the state veterinarian or notifying another public health official or department. 26% reported that they would perform additional testing, and 17% reported that they would notify their state agriculture department. 2 laboratories reported that they would submit the isolate to the Vet Learn surveillance program if a CRO was detected from a companion animal. However, these mechanisms were still under development at the time the survey was completed. 7 labs reported they would not take any additional actions if they detected carbapenem resistance. We also asked about awareness of public health reporting mandates for carbapenem resistance. Overall, 17 of 21, or 81% of labs surveyed, said that they were not aware of public health mandates that required reporting the identification of any carbapenem-resistant veterinary isolate to public health. Among the 4 labs that reported knowledge of public health reporting mandates for carbapenem resistance in their state, 2 were unsure if the reporting mandate extended to veterinary isolates. We investigated public health reporting requirements by state and overall found the landscape of applicable reporting mandates difficult to interpret because most are written in the context of human healthcare settings. This slide displays current state public health reporting requirements for carbapenem-resistant Enterobacterales as we understand them, as well as the reported awareness of public health reporting mandates for veterinary isolates among the laboratories surveyed. 20 states where surveyed laboratories are located have public health reporting requirements that extend to all clinical laboratories. However, only 2 reporting mandates explicitly mention veterinary isolates or veterinary diagnostic laboratories. For the 2 labs, O and T highlighted that expressed uncertainty about reporting mandates. The public health reporting guidelines for these states aren't clear if all clinical laboratories is inclusive of VDLs because, again, the context of the mandates is exclusively human healthcare. For the 2 labs, D and K highlighted that reported awareness of a mandate. State D has a reporting mandate that applies to all laboratories and explicitly mentions veterinary isolates. State K does not require reporting of CRE isolates according to publicly available information. To summarize, in this survey of VDLs, most, or 96% of labs, reported the ability to detect carbapenem resistance, but few reported having the capability to perform additional testing to detect carbapenemase production or carbapenemase genes. Additional testing to further characterize carbapenem-resistant isolates is important to understand their public health concern, as isolates that produce carbapenemases can rapidly spread multidrug resistance. As CROs can cause healthcare-acquired infections in animals, timely identification of these pathogens is critical to protect animal health and prevent spread in animals. Recent outbreaks of CROs in companion animals resulted in high attack rates among hospitalized patients. Delays in detection of these pathogens can result in increased transmission in veterinary settings. Further characterization of CROs is also critical to inform public health investigations to identify any possible link to human clinical isolates. A notable proportion of VDLs, 40% of those surveyed, identified at least 1 CRO from a companion animal during the time period of interest. And among these labs, the number of CROs identified varied. Although the overall number of CROs detected was small, these findings suggest that CROs might represent an emerging health issue for companion animals. Therefore, CRO surveillance would aid in describing the burden of CROs, risk factors for infection, transmission in veterinary settings, and potential impacts on animal and human health. These data and information are needed to make effective and actionable recommendations around testing, reporting, control, and prevention of CROs in veterinary healthcare settings. And displayed on the slide are links to 2 recently developed sets of frequently asked questions, one for veterinarians and one for pet owners about CRE, which includes information on transmission, infection control practices, and prevention measures. Recent outbreaks of CROs in companion animals illustrate that this is a One Health issue that can have a significant impact on both human and animal health. A multi-sectoral collaboration is needed to prevent the spread of carbapenem resistance among companion animals and people. However, most laboratories reported that they would not notify public health if carbapenem resistance was detected from a companion animal. Furthermore, some laboratories expressed uncertainty about the scope of known public health reporting mandates for carbapenem resistance as they apply to veterinary sources. As previously discussed, many states where surveyed labs are located have public health reporting mandates that extend to all clinical laboratories. However, they do not explicitly mention applicability to VDLs, leaving room for interpretation regarding these mandates, which again are often written exclusively in the context of human healthcare settings. Outreach by public health to VDLs could help clarify reporting requirements and expectations, as well as identify potential public health resources for testing and infection control in the event that CROs are identified. VDLs that have the capacity for mechanism testing and experience collaborating with public health could also share best practices for other VDLs looking to expand their testing capability. The findings of this analysis are subject to several limitations. First, laboratories surveyed were those affiliated with a state or university and frequently functioned as reference laboratories for veterinary referral settings. Thus, the findings of our survey are not generalizable to all VDLs. Expanding the survey respondent universe to include corporate VDLs, which might conduct testing for primary and tertiary veterinary care settings, would further our understanding of the capacity of VDLs in the United States to detect carbapenem resistance from companion animals. Second, we were unable to calculate the prevalence of CROs in companion animals among the VDL surveyed, as respondents reported ranges for the number of isolates that underwent AST and the number resistant to a carbapenem. Third, we used publicly available information to interpret public health reporting mandates for carbapenem resistance as they apply to veterinary sources, which is subject to misinterpretation as most are written exclusively in the context of human healthcare settings. Although most VDL surveyed were able to detect CROs with their primary AST method, few had capacity for additional mechanism testing or awareness of public health reporting requirements. Addressing these gaps is a critical step towards monitoring CRO incidents and trends in veterinary medicine, preventing spread of these threats in veterinary settings, and conducting an effective One Health response to prevent outbreaks and disease spread among animals and people. Stronger collaboration and information sharing between public health and veterinary medicine is critical to inform infection control practices in veterinary settings and to prevent disease and antibiotic resistance. And with that, I'd like to thank all those listed on the slide for their collaboration on this initiative, especially to the VDLs who took the time to participate in the survey. So thank you again for the opportunity to present the survey findings, and we'd be happy to take any questions.
Thank you, Dr. Waltenberg. We do have a few questions in the Q&A feature. I will read them off. This first question is regarding VDL responses. Is there any thought that the response rate was lower due to that time being around or during the second COVID peak?
That's a great question. And we did attempt to address this when we sent out survey responses by providing email reminders as we were sending out the survey and giving VDLs enough time to take the survey. We also did our best to, although it was a 54 question survey, make it as streamlined as possible since we know there were multiple competing priorities. But that is something to consider when considering the number of VDLs that responded to the survey. And we do hope to expand the survey respondent universe to corporate VDLs and others to try and get a better understanding of overall capacity of VDLs within the US.
Thank you. Second question, again, regarding VDLs. Did any of the VDLs report specific connections to public health lab counterparts?
That's a great question. So the one area where they did mention specific counterparts was when we asked about actions that would be initiated if carbapenem resistance was detected. Two of the laboratories specifically mentioned the VetLearn surveillance program, that they would send the companion animal isolate through that program. Others had mentioned notifying their respective public health official when carbapenem resistance was detected, but didn't specifically mention, the partner organizations, just that there was kind of that existing relationship and that they would be notified.
Great, thank you. The last question we have, again, if there are any other questions, please type them into the Q&A feature. Last question we have, and I'll read that off. Do we have any idea of what percentage of the CRO isolates have their resistance due to plasmids? I'm thinking of resistance being acquired through gene cassettes via other antibiotic exposures as opposed to CA being exposed to carbapenem?
That's a great question. I think for companion animal isolates, I don't have an answer for you in terms of the number that your percentage of the CRO isolates have their resistance due to plasmids. I do think that that's an area for additional research in terms of specifically looking at, for among the laboratories that detected a carbapenem-resistant organism, we in our survey had asked for the number, but didn't necessarily ask questions about the type of carbapenemase detected. And so I think there's a lot more to do in terms of characterizing what CROs have already been detected among the companion animal population. And I also think that potentially looking at this information that we have from companion animals in relation to human clinical isolates, whether that be, what are some of the mechanisms circulating in the region that we know about in human clinical isolates. Unfortunately, our sample size was a little bit too small to kind of look for trends in terms of geographic patterns, but I do think that comparing, what we know about human clinical isolates to what we found from companion animal isolates would be really informative, since we just have a much larger data set when it comes to human clinical isolates.
A couple other questions. Was there any correlation between animal infection and human infection that your study found?
So this study was specifically limited to isolates from companion animals, but kind of the one of the reasons that we, from a public health perspective, was interested in learning more about detection among companion animal isolates is because previous outbreaks have documented transmission between animals and humans regarding carbapenem resistance. And so this was kind of the survey was kind of taking it a step back to learn more about the capacity for veterinary diagnostic labs to detect carbapenem resistance in companion animals.
Excellent. Got a question from one of our international colleagues. Gives you a good day, very interesting PowerPoint. If you could repeat this survey once more, what would you do differently this time around? For example, any missteps to avoid or aspects to improve upon? And he specifically says that he needs to conduct a similar research project in Australia.
That's a great question. I think I think kind of the nature of these surveys is kind of once you have the data to analyze, you generate more and more research questions. I think one of the areas that I would be interested in is, going back to the laboratories that did identify carbapenem resistance and see, if they would be willing to either, share their isolate to do further testing on it to really get an idea of the different, potentially carbapenemase that we're seeing among companion animal isolates. So I think some additional laboratory characterization is probably what I'd be interested in, although that's quite difficult to do in a survey. I might also ask some follow-up questions about the nature of past interactions with public health, just to get a better idea of kind of awareness of public health being used as a resource in veterinary medicine. So potentially asking about, if they're aware of other interactions that they've been involved with public health, just to get a better sense of kind of the nature of that relationship.
We are going to try something here that we have not tried before in these CE conferences. I'm going to ask Dr. Buchholz to, instead of me reading the question, to ask that question verbally, if that's okay with her.
Okay, yeah, so I was wondering what the general response would be if it was reported to public health, what the general response by public health would be. And I guess it would vary, but kind of the general steps that would be taken if a veterinary diagnostic lab did isolate a CR organism.
Yeah, that's a great question. And I actually might ask my colleague, Dr. Nichols, to comment on this one, since she has been involved in some of these responses when we do actually at CDC receive requests from state health departments or local health departments to kind of help in responding to these instances when carbapenem resistance is detected from a veterinary setting.
Yeah, that is a good question. And we've encountered this a couple times. I think one of the primary things is to work with the veterinary hospital to try and do any additional case finding. So there's kind of two components. There's addressing the any ongoing transmission or issues at the healthcare setting. So in that case, the veterinary setting. And then there's also potential for some risk to human health. So there's some follow-up that's often done among animal owners, mostly to find out if there's anyone who is immune compromised, those who potentially work in healthcare settings to look at where the introduction of this might come from. And then also there may be instructions to follow up with either public health or their human healthcare providers to actually do some screening or to test for E. coli in this particular instance that might be offered. With respect to the veterinary settings, we've had veterinary clinics who've adopted kind of a couple different approaches. One, to really take it as an opportunity to work with their public health colleagues to do a walkthrough of their facility and look at infection prevention. We were talking with a colleague at a veterinary hospital the other day who, in the midst of something like this, actually found that very, very helpful. Oftentimes, our veterinary clinicians and practitioners are very familiar with our state animal health officials and maybe even familiar with our state public health veterinarians, but they haven't been introduced to the healthcare acquired infection epidemiologists or even colleagues at, say, a human hospital who work in infection prevention. And so having that two-way dialogue and having an opportunity to have someone visit and talk about cleaning of endotracheal tubes, or why it might not be a good idea to scotch tape something in an isolation ward to a door, how to clean stethoscopes, those types of things have been really helpful. And then there's also often recommendations if it's a referral center to make sure that you're looking at any patients who were infected or colonized to find out where they had been prior to coming into your facility. I will say in the instances that we've identified, at least one of them did have an ongoing outbreak that was detected after follow-up with the index patient.
If I can have Dr. Henenfent ask his question.
Hello, this is Dr. Henenfent. Can you hear me?
We can.
Yep. So I was just wondering if once a veterinary diagnostic lab identified a positive isolate, was it asked if they then performed whole genome sequencing and then entered the results into GenBank or another national database where then that sample could potentially be linked to other cases across species?
Thanks, Andrew, for that question. That's a really good question. We actually did not ask, what kind of the results were, what they did with the results from whole genome sequencing if they were performed. So few of our survey respondents reported performing whole genome sequencing, but I do think that's a good next step, or even potentially like a follow-up question to that if they would be willing, if they planned to upload that sequence to a national database, because I do think that would help get a better idea of what resistance looks like if it's something that had been detected previously, and get a better idea if they're seeing that in other isolates and how closely related it is to isolates that have been identified in the past.
All right, excellent. And one final question that we have in our question bank, Dr. Badada, can you ask your question, please?
Yeah, can you hear me, please?
Yes, we can hear you.
Yeah, thank you so much for the marvelous presentation. I really like that one. I do have very simple questions just for curiosity. My first question would be, what were some of the major contributing factors for the occurrence of this drug resistance? I mean, it's coming to the public nowadays about people are starting talking about those drug resistance. It's becoming a very concerning issue worldwide even. So what are some of the major contributing factors for the occurrence of drug resistance, especially with these antimicrobials? in vets as well as human medical setups? And maybe do you have a similar plan to study like you did for pet animals in other domestic animals, like large animals in equine, some other animals? Thanks so much again.
Thank you for that question. I think that's actually one of the kind of follow-up questions that was generated after we saw some of the responses from the survey. So we, were a little bit surprised at the number of resistant organisms that were identified over an 18-month period. And so I think part of what we mean by further characterization of these isolates is not only additional laboratory testing, but also to gather more information about, risk factors for transmission among these animals. So, gathering more information when carbapenem resistance is detected to learn more about what some of these reasons or contributing factors were for the occurrence of resistance. And so I think that kind of just building up that knowledge base will really help in terms of developing guidelines and prevention measures that can be implemented in veterinary settings. So I do think that it's two components: additional laboratory testing, and then also potentially looking at some other risk factors for infection and how that can be translated then into preventative action. And at this time, we don't have current plans to conduct a similar survey for other species, but we do have plans to increase the survey respondent universe specifically for companion animals by including some of the corporate veterinary diagnostic labs.
Thanks, Michelle. And I will add, it's interesting when we see these outbreaks and there's discussion. I think each time presents an opportunity for those who are investigating to potentially ask some of these. On the human side, I know when there's a kind of a One Health approach to this, the epidemiologists on the human health or public health side might be able to bring some of that experience to questionnaire development. And I know in some of these investigations, there has been a significant interest in previous carbapenem use, because in the human healthcare side, one of the main factors associated with carbapenem resistance is prior use of carbapenems. There's also been other factors described, such as other antibiotic treatment and very long duration, ventilator-acquired pneumonia, presence of some intravascular or indwelling devices, but the contribution to infection remains unclear. And it's tough to apply that to our veterinary settings, especially because we don't believe that the carbapenems are often used. And so having prior exposure leading to the development of resistance might be a little less common than we'd see on the human side.
I will go ahead and read this question. Was there a geographical trend for recognition of CRO?
Yeah, so we didn't necessarily see the recognition of CRO specific to any one census region. I mean, we did have, 40% of the surveyed laboratories who identified at least one CRO, and they were quite widespread throughout the US. So we didn't necessarily see any clusters in any census region. So I would say, yeah, overall, we didn't see any clustering geographically.
Questions keep coming in. Dr. Josephine Afima. Dr. Afima?
Yes, I was just wondering what other resistances are associated with CROs?
I might say, I don't think we necessarily know this from the companion animal perspective. I don't know, Megan, if you know from a human clinical standpoint, what other resistances are typically associated with carbapenem resistance among humans.
I don't know them off the top of my head, but I do know that there has been some publications recently that talk about the different genes that they find among NDM5 E. coli organisms and what type of resistance those genes also confer. So I don't know those right off the top of my head, but I would say there are some publications in the literature, especially in a lot of the international literature, that might have some of those answers.
Hey, I believe we have come to the end of our questions, unless somebody else types one in real quick here. Doesn't look like it. So what I would like to do is just close out with a few announcements. first of all, I want to thank our speakers, Dr. Nichols and Dr. Waltenberg. Excellent presentation. I want to also just remind everybody that CE certificates, we will get those out as soon as possible. We have to verify membership in our partner organization. So it does take us about a week or so to get all that done. So you should see something in your email here within the next 7 to 10 days. I know there are a couple phone callers on the line. If you can just email me at admin@acvpm.org, your name that's tied to the phone number. That way I can make sure that I can get you a CE certificate. And we will close out. I will ask Dr. Tomasi if she has any closing comments.
Thank you. No, again, as my regular reminder, if anyone is interested in presenting or has a topic they would like to hear, please let the CE committee know. And we'll see about getting that presenter. We are still looking for a November speaker for 2021, and all of 2022 is still open. Thank you very much.
And I will add, again, you can email me and ask questions about how to apply to be a speaker. I will help you out because we're always looking for speakers. We can't make this program work without our volunteers. So once again, I thank everybody for their attendance, and I thank our two speakers for doing such an excellent job. Everybody have a wonderful afternoon, and hope to see you at our CE webinar next month. Thank you.